Hi, Mrs. Meltreader. I've got your first set of notes for Chapter 3 on macromolecules. So we're going to be talking about what makes us up and what makes up our food. So the first thing is that we're talking about carbon-based life forms. And if we're looking on another planet, we're not sure if it's going to be carbon-based life forms or not. But that's why we say carbon-based life forms, because pretty much everything that makes us up has carbon in some way, shape, or form. So we'll kind of go through that. So carbon is the framework of all of our biological molecules. It can form up to 4 covalent bonds, and we remember that because it is needing those 4 valence electrons, right? So it can form those 4 bonds, and when it forms those bonds, it's going to do it in the shape of a ring, or maybe a chain, or a tube, or a coil, or even some other different shapes, but those are the main ones. One thing that carbon is really good at forming are things called hydrocarbons. And we talked about how with those 4 valence electrons, it likes to grab onto some hydrogens because hydrogen wants to share its electron. So hydrocarbons have lots of energy stored in them. Those are going to be things that are like a lipid form. So they're going to have lots of energy, be a good fuel source. They're made up of only carbon and hydrogen. And of course, because they have that equal sharing, they're going to be nonpolar. And it's their functional groups of those hydrocarbons that aren't specifically lipids that will make up its properties. Okay, so let's talk a little bit more about those functional groups. So you don't have to memorize all of these, but they are things that will come up when we're looking at our different structures and our different macromolecules. OH, the one on the top here, is called the hydroxyl, and that one's pretty important. We'll see that hanging around a lot. We'll see those OH groups hanging around a lot. We'll also see the carbonyl groups where there's a C double bonded to an O. We'll see that a lot when we look at different molecules. And then we'll see the phosphate group, especially when we talk about DNA and ATP. And we will see the amino groups when we talk about the proteins and amino acids. Okay, so it wouldn't hurt to have those memorized, but I'm not going to test you on them. Okay, so sometimes we have things called isomers. Okay, and an isomer it's the same molecular, so C6H12O6, or empirical formula. That's where we take CH2O to like the nth power, like 4, we could say, which means we'd have 4 carbons and 8 hydrogens and 4 oxygens, that kind of thing. So they have that same molecular basis, okay? Similar to another word that we heard about called isotopes, but not the same thing, so don't get those confused. Isomers are compounds that have a similar, or the same, molecular formula, except there's a difference in their carbon skeleton. Okay? And we have like glucose and fructose. Those are isomers. They are both C6H12O6. We like to use glucose. Plants produce fructose. Okay, so when we have those isomers, there's some different kinds or different groupings of those. And the differences is, I'm stumbling today. The differences is in how they are attached. If that is the only difference, then they're called stereoisomers. Okay. If those groups happen to be attached in a different way, but they're mirror images of each other, like in our diagram here, where we have W on the left and Y on the right, and here we have Y on the left and W on the right, Those are mirror images. Those are called enantiomers or chiral molecules because of how they bend light. And they bend light in a way, two different ways. If they bend it right, that's called dextro. And you won't need to worry about what specific ones bend that way. I'm just telling you these things so you know that there are different types. Okay, so right is a dextro or D, and left is levo, Latin for left, and would be L. Okay, most organisms are going to produce one or the other. They're not going to produce both the D and the L form. So in our book, it talks about that our bodies produce primarily D, so dextrose sugar, so right-handed sugars, and L amino acids. So take a minute, pause the video, see if you can identify what these would be. Okay, if you said these are enantiomers, you would be correct. And alpha glucose and beta glucose are stereoisomers. Good job. Okay, so during our time, we're going to talk about 4 basic macromolecules, and those are carbohydrates, proteins, lipids, and nucleic acids. Usually these things are polymers, which means they are longer, bigger molecules that are made up of little smaller units that we call the monomers, the subunits. So I like to think about it as each individual Lego would be a monomer, and the structure you build out of the Legos would be a polymer. Or you can think of it as beads on a necklace. So each pearl would be a monomer, but the whole necklace would be a polymer. So polymer means many, okay? And we'll talk about what each of those monomers are as we go through the different macromolecules. So here are some common ones. We have our carbohydrates, like starch, cellulose, chitin, okay? And those have subunits of glucose and slightly modified glucose. We have our nucleic acids, our DNAs and RNAs, and their subgroup are nucleotides. We have our proteins. There's 2 main kinds, functional proteins and structural proteins. We'll dive a little bit more into the differences later. And their monomers are amino acids. And then finally, we have the lipids, like triglycerides, phospholipids found in our cell membranes, and steroids, prostaglandins, and I can't see that last one. Oh, terpenes. Those are found in plants. Okay. So just another little image of the different macromolecule, the polymer, and then its monomer. So go through. Okay, so this is the last little piece for this video, and that is how do we put these things together? And this goes for all of these polymers. So it's a process called dehydration or condensation. You may see both, okay? Dehydration means to dehydrate or to remove water from it. So what happens is it's going to remove an OH group from one monomer, one piece, and an H from another piece. And if you take an H and an OH, you get H2O or water. And it makes this covalent bond magic, and voila, we have water. Okay, this process is carried out by different enzymes. And the one thing that's super important here is that one H2O is removed for every bond. So if you're linking 2 monomers, 2 glucoses together, that's one bond. Okay, so 1 water will be removed. If you're linking 4 monomers together, 4 glucoses, you have 3 bonds. And it's always the number that you're linking together minus 1. Okay, if you're linking 54 monomers together, you would have 53 waters that are produced. Okay, if you're linking 1,600, so 1,600 monomers together, you would have 1,599 bonds. So that's just how you go. So if it was asking you how many waters, it's always how many molecules are you linking together minus 1. Okay. And we'll learn a little bit more about that process of each one as we go through them. But it's called dehydration, and it's putting two monomers together by removing a water. Okay. When we take them apart, so if we eat some sucrose and we have to break that down inside our body, we have to take that sucrose and break it down into all of the smaller glucose molecules so that we can shove it into our mitochondria and get energy from it. So that process is called hydrolysis. Okay, so putting them together, it's called dehydration. Taking them apart is called hydrolysis, and it's where we're going to add water in. Okay, so in order to do that, an H is added to one subunit, and a hydroxyl or an OH is added to the subunit next to it. And you get, since you've added two H's and an O, you've added one water. Okay? And it's going to push right in there and separate those monomers. So the number of monomers minus 1 equals the number of waters to be added. So if you have two monomers, then you take one away, so you're going to add 1 water. If you have 3 monomers, you would take 1 away, so you'd add 2 waters. 4 would be adding 3 waters. Okay, so again with that bonding, so it's just in the reverse process. So that ends our talk for today on carbon-based molecules and how we put them together and take them apart in polymer form. See you later.